Okay, the next section is on protein metabolism. So we can break down our proteins into their monomers, which are amino acids, and either reuse those amino acids to make new proteins, or we can even use the amino acids as a source of energy. So our proteins are constantly being broken down and being replaced or being built back up because proteins have a limited lifespan. As we break down the proteins, their amino acids aren't just excreted, we actually recycle them and use them to make new ones or modify some other compounds that can utilize them. Proteins are not stored in the body, so when dietary proteins are consumed in excess, we actually will take those proteins and oxidize them as a source of fuel for energy, or we can even take excess proteins and convert them to fat for future energy needs. So the goal of amino acid breakdown is basically to produce molecules that can be oxidized for energy in the citric acid cycle, or to convert these into glucose themselves. But before we can actually use amino acids and before they can be oxidized, they have to be deaminated. And deaminated is referring to the process of taking their amine group, which contains a nitrogen. So amine group is NH2. We have to remove that. And basically, after we remove the amine group from an amino acid, we can convert that product into pyruvic acid or to one of the keto acid intermediates of the citric acid cycle. So we have three events of amino acid degradation, the first being that transamination. We also have oxidative deamination and then keto acid modification. So in transamination, we have a lot of amino acids that can transfer their amine group to an alpha ketogluteric acid, which is a citric acid cycle keto acid. And when we transfer the amine group to this acid, we transform it into glutamic acid. In the process, that original amino acid becomes a keto acid. It has an oxygen atom where the amine group originally was. This reaction is also reversible, so we can reform the original reactants if needed. The second step is going to be oxidative deamination, and this is going to occur in our liver. The amine group from the glutamic acid that we just produced from transamination is going to be removed. And when we remove it in the form of ammonia, which is NH3. And we can regenerate the alpha ketogluteric acid. Our ammonia is going to then combine with carbon dioxide to form urea. The urea is going to be released to the blood and excreted eventually in our urine. Because ammonia is toxic to our cells, our glutamic acid is going to take that amine group into the urea cycle, and this is a really important process because the urea cycle helps rid the body of our toxic ammonia. All right, the third step is keto acid modification. So the keto acids that we got from transamination are altered as necessary in order to produce metabolites that can go into the citric acid cycle. The most important metabolites are going to be our pyruvic acid, our acetyl-CoA, our alpha-glutaric acid, and our oxaloacetic acid. Because the reactions of glycolysis are reversible, deaminated amino acids that are converted to pyruvic acid can be reconverted to glucose and help contribute to gluconeogenesis. Remember, that's the process of making glucose from something that wasn't a carbohydrate to begin with. So just a couple terms and an image for you. When we're talking about transamination, that's the process of taking an amine group from an amino acid and transferring it to an alpha ketogluteric acid. And we're going to generate glutamic acid once we attach that amine group to alpha ketogluteric acid. Oxidative deamination is referring to the removal of that amine group, And when we move the amine group, we remove it as ammonia, and we're able to regenerate that alpha ketogluteric acid. So we have something called the catabolic anabolic steady state. This is a dynamic state in which our organic molecules, except for DNA, so basically our macromolecules, are continuously broken down and rebuilt. And then our body uses nutrient pools, which are our stores of amino acids, carbohydrates, and fats. And these pools can be converted to different things because they are all linked via a common intermediate. And the amount and direction of conversion are going to be directed by our liver, how much fat we have or adipose tissue,